And so it says choose the best answer or best match, and all answers will be chosen. So this is basically just a vocabulary question. So it's probably one that you can get just by looking through your notes. But we can go over it a little bit. So codominance and incomplete dominance are the ones that I think most people messed up on because they flipped them around. But remember, codominance is when we're displaying things simultaneously. And that's like our spotted cows and stuff. So they're coexisting. Both of the phenotypes are there at the same time. Whereas with incomplete dominance, you have a blending of the two. So that's like our white and red flower making a pink. It's been like an incomplete, it's a blending or a merge of the two. Okay, so those are kind of what you're looking for there. Polygenic traits should be pretty straightforward. Poly is many, genic is genes, so many genes make up a trait. So that's what you're looking for in your answer there. And epistasis is when we have one gene that's affecting another gene. Maybe it's masking it, maybe it's amplifying it, whatever it is that it's doing. But that was like our Labrador retrievers, where we had the coat color that was affected by the amount of melanin produced through another gene. Okay, so those are sort of what we're looking for there. So, if we were to pick our answers, it would just be that, like this one, and more than two, more than one gene effects. Okay, so that's what you're looking for there. Question 2, okay, we mentioned this one. I had already had the answer from one of the other class periods. It says that the diploid number of chromosomes is 64. Which of these represents the correct number of chromosomes as we go through this whole cycle? And so the thing that I think gets most people is they think that it starts with 128, but it doesn't. So when you're thinking of 128 there, you're thinking of the number of sister chromatids as opposed to the number of chromosomes. So we do duplicate the amount of genetic material that we have, but that doesn't give us more chromosomes. It just gives us sister chromatid copies. And so the chromosome number after G2 is still technically the same. It's still 64, just like it would be in a normal non-dividing or going through the cell cycle cell. And the other big thing here is that we know that after meiosis, we are going to have haploid gametes. And so we have to have the number of chromosomes, so 32. And then after fertilization, we have to go back to our diploid number, which means that we're going back to 64. So even if you weren't sure about the whole chromatids versus chromosomes bit, the thing that should sort of tip you off is that the reason why you wouldn't pick this 128 to 32, is because after fertilization, you should be a diploid organism. So you should have 64 as an option. And then that means you can also rule out this one here because you're not going to go from 128 to a fourth of that total. You don't lose 3/4 of your chromosomes when you're doing this. We only end up having our numbers. Okay, so that's question 2. Question 3 is about sea turtles. This one is actually just about interpreting a graph. And it's something that we'll probably be doing a little bit more as we go on because it is an important skill. And so this is basically saying that we're measuring how the temperature affects the percent of females in the turtle population. And so the answer here is going to be what describes the results is that warmer nests are producing more female sea turtles than do cooler nests. So we can notice that as the temperature is increasing, so too is the percentage of females. So you can get this one even without the genetics knowledge per se. The other ones that you could rule out if you couldn't just do that. So it says sex ratio of turtles is genetically determined. We can see here that temperature is what's affecting it. And so even if there is some bit of genetics, you can't know that based off of the graph, because the graph has nothing to do with its genetics. So you can't make that assumption based off of the results shown. And then female sea turtles search for cooler beaches in order to have more male offspring. You can't know that female sea turtles do that. It doesn't say that, like, you know, they react to different temperatures and go different places. It's solely that we have more female sea turtles when it's warmer. So animals, generally speaking, are not going to recognize that they need more male offspring and then go to a cooler beach. They're not that advanced in that regard often. And then the percentage that survive to adulthood, again, there's nothing about survival in here, so it can't be that. So question 4, which I have gotten a couple times, says, bananas are usually diploid organisms. Which of the following best explains why triploid bananas, so 3N, cannot produce seeds? And so the best way to kind of think about this is with a picture, I think. So, you know, normally we have these diploid organisms. They're going to have the 2 chromosomes, you know, our 2 homologous chromosomes, chromatids, all that type of stuff. And these are going to be separated into gametes. And then those gametes are going to come together in fertilization to, again, make that 2N, that diploid individual. But if we have 3 when we're normally supposed to have 2, that's going to cause a lot of problems in terms of developing those gametes, because you can't just your cell doesn't just cut a chromosome in half and make it 1 1/2. And even if it could, that would cause a lot of problems in and of itself. So you can have all kinds of weird combinations where you could have like 3 going to one cell and 0 going to the other, or, you know, 2 and 1, and then that 2 merges with another 2 and makes a 4 ploidy cell. So your body, because it has those checkpoints, is basically not going to allow that to happen, because it doesn't want to make these jacked up gametes. And so because of that, the answer is going to be that the production of gametes is disrupted because of the unequal pairing of homologous chromosomes. Okay? And you can probably rule out some of the other ones as well, like banana plants lacking genetic diversity. You know, when we get an extra copy of a chromosome, that's not lowering the amount of genetic diversity that we have. Production of seeds is not required because they produce gametes without fertilization. You produce gametes without fertilization anyway. You make the gametes and then they fertilize, so this is irrelevant. And then the cells of the banana plant are unable to replicate DNA. This, you know, we can still replicate DNA. It doesn't prevent that from happening. So this third answer is the only one that could really make sense. Okay. Did we have 5 from anybody? Yeah, we did. Okay. 5. So 5 is a standard, even though it's got a lot of stuff to it, this is just a dihybrid cross. So we take true breeding plants, we breed them together, we get an F1 generation that's all heterozygotes, and then we cross those heterozygotes. And this is a pretty big hint here. We have 9 times as many plants with yellow round seeds as plants with green wrinkled seeds. So that should make you think of our 9-3 to 3-1 ratio, because this is a dihybrid cross. So from either of those things, you should have recognized that. And then it says, which of the following would Mendel say best explains these results? And so the answer here is going to be that they assort independently. So you could have gotten this through a number of means. One is that when we discussed dihybrid crosses, we directly said that the results of a dihybrid cross were what gave us the law of independent assortment. But, you know, if you don't recognize, you know, if definitions and vocab and stuff are harder for you, you can also kind of rule the other answers out too. So gene segments on sister chromatids cross over. This does not occur. Remember, when we think of our chromosomes, so our chromatids and our homologous chromosomes, we have those sets, right? And homologous chromosomes are, you know, we have 1 here and 1 here. And they come together and they cross over. But sister chromatids don't cross over with each other. It's when the homologous chromosomes come together that they cross over with each other. So this doesn't happen at all. The allele pairs of each parent stay together. So this, again, if you recognize the 9 times and you recognize that ratio, you know that we have those 2 sets of 3 where we have recombinants, where we have those heterozygous phenotypes. And so if this was true, if the alleles always stayed together, we would never have those, one dominant, one recessive answers. So we can rule this out. And then finally, alleles that are on non-homologous chromosomes recombine. Again, homologous chromosomes are what are going to cross over. If we take chromosome 1 and chromosome 23 and put them together, they're not going to cross over. They don't do that. So that's why you could rule all the other ones out, even if you didn't remember the exact, relationship. Okay, and then that takes us to 6, which someone just asked me about. So we're going to take a look at this, because 6 and thereby 7 are probably 2 of the most involved ones, just because you have to, kind of tackle it logically. And so to help facilitate this, I did a crappy little drawing here that has a ton of erase marks and stuff on it, because I keep using it over and over again, but it still fits the purpose. So we have this pedigree here, and we have to determine whether it is autosomal or sex-linked, and dominant or recessive. So the nice thing about this one in particular is there's a really big tip-off that could get you the answer incredibly quickly. And that is if we look right here, where we have 2 parents that do not show the trait, and yet one of their offspring does. So because of that, we can instantly say that it is not going to be dominant. And you can check that really easily, because these would both have to be double small allele, and this person has to have a dominant allele if they show it. And so this is impossible right off the bat. And it doesn't change if you make it sex-linked either. So do this and then change this to a Y. They would still have to have an X uppercase A, and that can't be given by either parent, because they have to show double recessive alleles, because we are assuming that it's dominant. Okay, so we can instantly rule out both dominant options, which leaves us with only the question of whether it is autosomal or sex-linked. And so one of the things that would be a big tip-off for me as to why it's probably not a sex-linked is we're not seeing that, because we know it has to be a sex-linked recessive, and we've talked about with sex-linked recessives that generally the males are going to get it and the females are not going to get it, but we can see in multiple different cases that female individuals have it, like here and these 2 down here. So we could probably rule that out, but if we wanted to be sure, we can also look at sort of this first bit here. So if we are looking at, we're pretending that it's an X-linked recessive, then we would say that this person would have to have 2 lowercase alleles, and this person would obviously have to have an uppercase because they're not showing it, and the Y is just kind of that, crappy chromosome that doesn't matter. But because of this person can only possibly get 1 recessive allele, because the other X that they're getting is from their father, which is dominant. And if that's the case, this person couldn't have the double recessive that they would need, and so we can safely rule that out, which means that the answer has to be autosomal recessive. Okay? So there's a number of different indicators that can kind of get you to this quickly, and there are ways to check if you want to be 100% sure that is actually the case. Okay? So that answers 6, and then 7 was saying, let me go back to it really fast. 7 was saying, what is the probability that individual 3-3, and of course, even though I put it underneath it in the editing bit, it did move over, but I mentioned which one it is, so the leftmost unshaded circle in 3rd row. So that means this one here, underneath the parents that are not showing the trait, so this circle down here, what's the chance that it would not express the tracked trait? And so essentially that's saying, what's the chance what was the chance that it was an unshaded one to begin with? You know, did it get lucky or not, basically? And so what we can do is that in this case, actually, the sibling kind of gives away the percentage, because you can't actually calculate an exact percentage for this. So we came to the assumption that it's autosomal recessive, which means that, so this is going to be A, because it's expressing the trait, and we know that it's recessive, which if we do that, we actually know what the genotype of the parents is, because these parents both have to be then heterozygotes, because they don't show the trait, which means they're not double recessive, but they need to both be able to contribute a recessive allele. So if even one of them was double dominant, if we did our cross, we would have big A, and then big A, small a. All of these would be unaffected. None of them would be shaded in. And so we know what these have to be, which means that we can do a cross to determine what the chance of getting this is. So now we do our heterozygous cross, which you're probably familiar with enough at this point that you don't even need to do it. But we can see that there is a 3 out of 4 chance that we have at least one dominant allele and therefore do not show the trait. So there's a 75% chance that we don't show the trait, 75% chance that we get this result here. So they had a pretty good chance of not developing the trait. Okay, so that's 6 and 7. And if we do have time at the end and you need any additional help with it, I can go over it a little bit more again, but I want to get to the rest of the questions. So I know we have 10 and 11, 10, 11, 13. Okay, 10, 11, and 13. So 10, this was the Pikachu one. And so this is basically just, you have to do 2 things in this problem. Yeah, I can do 12 as well. You have to do 2 things in this problem. You need to identify what the recombinants are, and then you need to do the equation that I gave you. So from this, we can see that we mated a wild-type Pikachu. So they had yellow skin and red cheeks, and an orange Pikachu with pink cheeks. So this is the mutant Pikachu. And so when we put those, so those are our parental genotypes. And so their offspring that are either wild type, which again is the one with yellow skin and red cheeks, or the orange pink, those are parental. So those are not recombinant because we're seeing the same combinations that we had from the parents. There's no interlap between the dominant and recessive alleles, or phenotypes, I should say. Whereas these yellow, pink, and orange, red, they're both getting one trait from each parent, which means that there has to have been some kind of crossing over event. And so this 15 yellow, pink, it has one dominant and one recessive, and then 10 has one recessive and one dominant. So these are our recombinants. So the number of recombinants here is 25. And then you're going to take that 25 and divide it by the total number of all of them, and then multiply that by 100. Okay, so that's how you do that answer. Okay, and I'm not going to do the math right now, but that's the setup for it. So 11, it says a farmer crosses 2 plants and notices that half of the offspring are tall and half are short. Assuming that this follows basic Mendelian genetics and is related to one gene, what does this indicate about the parents? So we're doing a cross, and you find that half are tall and half are short. So one way that you could, well, probably the best way to do it. What does this indicate about the parents? So half of the offspring are tall and half are short. So that 50-50 ratio is a pretty good indicator of what it's going to be, because we know that our options are that we have big T and little t for height, and we don't have like a 1 to 2 to 1 ratio. We don't have like a medium height thing, so this isn't going to be like codominant. So this is going to be a standard Mendelian thing, and so our results we have to have 2 out of 4 are going to be short, and the only way you can get that is with recessive alleles. So we're going to have T here. And then, what did I do wrong? What am I doing? Then that would be T, and that would be T, which we don't. I've been dumb. No, this would be. Hold on. The other class period didn't ask this question, so I didn't do this. Okay, about the parents. One is heterozygous and one is homozygous recessive. That's right. Okay. Yeah. So heterozygous. So T. So one is going to be like this, and one is going to be like this. Because if we do that cross, we're going to get. Oh, I see. Yeah, I was just being stupid. My bad. Yeah. So we can do this cross. And you could do it in the way that I was doing it earlier, where you start with those two. I just did it, going to the side instead of up and down. But you would have one of them would have to be a positive or a dominant, because we need to have a dominant, and then the other would have to be a recessive. So whichever way you ended up setting it up. But either way, you'd need to have half being tall and half being short. And so that's the way you could do it. So realistically, you know right away that 2 of them, so if I were to re-explain this in a less crappy way, 2 of them have to have 2 recessive alleles in order to be short. And if 2 of them have to have recessive alleles, there's obviously going to be some recessive, but we know that some are tall, so we know that there's going to have to be at least 1 tall allele, and so that's going to give us the heterozygotes, and so you can fill in the rest from there. Okay, so you're basically just kind of working backwards in that one, but at the end of the day, it's the same. And the nice thing is, if you weren't sure to work backwards, you could also do a test for each of these and see if you got the right thing. So, if I wasn't sure, I could do a heterozygote cross with the homozygous recessive or any of the others, and I could see if you get the right percentage. So there's a fast way to do it, which is the working backwards, and there's a slow way to do it, which is just testing each one individually. 12. So which of the following is not true about meiosis? So this one, you can just rule out things, or you can automatically recognize that homologous chromosomes are not separated in anaphase of meiosis 2. So we did talk about how homologous chromosomes are separated, but that happens in meiosis 1. So remember, the order was that we separate the homologous chromosomes, and then we separate the sister chromatids, which makes sense because we have to take, the bigger number of chromosomes and separate those, and then the smaller number of genetic material and separate those chromatids, okay? And all the other ones do occur or are important for meiosis. Even this one where it says it requires growth and DNA duplication, we still have to grow and duplicate in order to split. So it's not any different from mitosis in that regard. And then 13, this is having to do with the multiplication rule that we talked about on Monday. And so I gave you the hint that told you to use the multiplication rule and to break it down into parts to give you a little bit more. And essentially all this comes down to is we're looking to see the chance that based off of Mendelian rules of dominance, what's the chance that our offspring is going to exhibit all dominant phenotypes? So AKA, what's the chance that all of those genes are going to show either the heterozygous phenotype or the homozygous dominant phenotype? And so you do a cross for each of these. And so a heterozygous cross we even did in one of the last problems. That's going to give you, if we do it over here, heterozygous cross is going to be a 3-to-1 ratio. So 3 of them are dominant and 1 is not. So this is 3/4. And then the only other one that matters, because In both B and in C, you can't ever get two recessive alleles because, you know, this one has a double dominant and this one has a double dominant. So you can ignore those, and that only leaves D, which when you do the cross for D, that gets you 1/2. And so 3/4 times 1/2 is going to be 3/8. And so that's the answer for that one. Okay, and that takes us to pretty much the last minute here. We do have office hours, like literally right after this. So if you do want to stick around so that I can answer more of your questions or whatever, you can. Otherwise, you're not obligated to be here anymore. So you can head out. And if you do, there's no homework for this weekend, but just be ready to jump right into it on Monday. And if you're headed out, then thank you, and I will talk to you on Monday. And have a nice weekend.